First of all, I would like to offer my salute to Goddess Saraswati, who is considered to be Goddess of knowledge in India. And we are here because we are so-called knowledgeable persons. Then I offer my salute to these great two personalities. One is Pandit Madan Mohan Malviya ji, who has created the world-class university known as Banaras University in the city of Varanasi. So I offer my salute to him because I am serving there as a professor, and it is possible because in the university would have not been there, probably I might have been somewhere else. And then next personality is Professor Mishra, who is considered to be the father of pharmacy in India, because he started the pharmacy education in India from this department of Pharmaceuticcs in 1932, whereas the university was established in 1916. So we are celebrating the 30th year of the Banaras University. My work is focused on basically a new technology which is known as a Pastillation technology. And this technology is basically being used in the chemical and agrochemical industries to convert hazardous dusty chemicals into the solid particles so that the hazards around the working environment of the chemical and agrochemical industries can be minimized. So first time we have tried to use this technology into the pharmaceutical formulations, and therefore I am just going to present my work. And before that, I would like to thank the organizer and also the chairman who has provided me the opportunity to talk on this dais, and also thank you as well as salute to you because you are here to hear my lecture. So the basic component of my research was lipid-based multiparticulate drug delivery system we have designed. And the drug we have taken as a doxorubicin is a relatively newer drug in the treatment of asthma and COPD. And the experimental work and the design and development which has been done with the pesticides was in relation to the development of 3 different formulations. One was for the immediate release, for having better and immediate efficacy, and then sustain and control release, and third was the pulsatile release. So pesticides were utilized for preparing 3 different types of the formulations, one for IR, another for CR, sustained release, and third for the pulsatile release, especially for the treatment of early morning attacks of the asthma. If patient consumes medicines in the night at the time, the sufficient concentration or the amount of the drug should be there early morning to treat the patients to counter that early morning effects of the asthma. And then we get the in vivo studies, pharmacokinetic as well as the gamma centrifugal studies. The first part, lipid-based multiparticulate system, and the literature till now, we have the systems which are available: nanoparticles, microparticles, granules, pellets, and beads. What is pesticides and what pastillation technology is? It's a widely used technique in chemical, as I said, petrochemical and agrochemical industries. It is used as for the solidification of dusty hazardous powders of chemicals into pesticides. And what are the pesticides? It is a hemispherical solid units of uniform size, which eases their handling as well as doesn't allow to be spread into the environment as far as the chemical and other industries are concerned. In this process, what is actually being done? The drops of the chemical substances in the molten state are deposited on the cool stainless steel surface for rapid solidification to generate the pesticides of uniform dimension. So we have just utilized this concept. This pastillation in chemical and agrochemical industries are done using a very big equipment known as a rotofarmer. In our lab, we have designed a very simple, innovative, a small unit by which we can design or we can develop and prepare the pesticides. So depending upon the size of the drops, the physical properties of the pesticides can be manipulated, and these certified droplets, which are called as a pesticides, may further be either packed as a capsule or maybe in the bottles, or sometimes even maybe, if possible, can be given as a suspension. These are rotofarmer which I was talking about. What is the concept behind pastillation? The chemicals, hazardous chemicals in the molten state, it is pumped, it is then being dropped through a nozzle on a cooled bed. It is a cooling surface of the bed, and this molten base is converted into the solidified mass, which we are calling as a crystals, and they are getting collected here. So this is what is being done in the chemical industries. Why pastillation in drug delivery system? As I am telling that this is the technology which we have used first time in pharmaceutical formulations. It is having a lot of advantages as it is suitable for a hygroscopic drug, for processing the ingredients, as ingredients are absolutely free from the use of water, it's environment friendly, stable and highly uniform in size because of these uniform size of the drops. Can be the variable size range 1 to 30 millimeters. They are having one high bulk density, better packaging properties in powders, and therefore ideal for handling, filling and packaging. Conversion of bulk molten liquid directly into the individual solidified units provides dust-free working environment, which I have already told. It's a single-step process, reduce the energy cost due to absence of number of the processing parameters of the formulations, and also it's very easy to pack the pastils for consumption to the patients and for going to the hands of the patients. The drug we have taken, so pastillation technology is the main focus in our research. Drug we have taken doxorubicin, as I said, for the asthma treatment, for immediate effect, sustain and control immune effect, and for nocturnal asthma, the early morning attacks through the versatile formulation. This is the profile of the doxorubicin. It was recently 2011. It was in India only 1 or 2 products were there. Now there are lots of products are there. Those in tablets in 400 mg of IGA immediately is given 2 to 3 times daily. These are the pharmacokinetic profiles. Its absorption is good, 62.7%. These are the other pharmacokinetic profile which is required for before going for the design of any formulation. I'll be going a little faster because a lot of material is there for showing to you, and since this is a new technology for pharmaceutical formulations. So our objective of the research was to explore the pastillation technology to design and perform technology for the development of novel and unique modified laser delivery system. And as I said, we designed immediate and controlled release. We designed the versatile release, and also we performed in pharmacokinetic and scintigraphic gamma scintigraphic studies to prove our hypothesis, what we have done in this work. This is what I was talking about. It's a very simple device, which we have developed in our lab. This is a metallic tube, is covered by ceramic insulator, and then this is there is a heating coil inside this insulation that is connected with the transformer. The drug is placed into the beds, maybe liquid beds or the PEG beds, put into that mixture. That mixture is put into this, dissolved into the molten state, and then this is connected with a syringe, and that syringe, through that syringe there is a plunger which is pressed to force the molten base, and through the needle this molten mixture is coming on the surface of this stainless steel plate, and before to that it is a lot of ice is placed just to make it a cold. Temperature can be varied by just keeping the variable amounts of the contents of the ice. And that what is the variable into this formulation? One is the temperature of this stainless steel surface. We try to keep two temperatures, 4 degree and 25 degree. And then height between the end surface of the needle and the upper surface of the steel plate. This is the second variable. And third is the I'll be showing you in the next slide. And this is the after dropping the particular shape of the pastils which we are getting like this, hemispherical pastils. And the contact angle is very important parameter. To the response surface plots, we have tried to see that with the three important variables, formulation variables in this case, third was the needle dimension. We have used two sizes, 16G and 20G. Dropping height is 1 centimeter, 3 centimeter, temperature of 8 was this. With Minitab, we just try to find the optimized parameters with the help of we measured the contact angle. With the help of the response surface plots, we concluded. So any two variables were measured for these response parameter was the contact angle. So for example, there is effect on radial size in dropping height and contact angle, effect of temperature of plate and dropping height of the contact angle, and effect of radial size and the temperature of the plate on the contact angle. And our results were, and if you see this is the, if you see the contact angle data, 105 to 125 degrees is considered to be a good flow property. And this particular contact angle we found with well optimized parameters like, when the radial dimensions were 2G, dropping height was 1 centimeter, and temperature of the plate was 4 degrees centigrade. So in our future formulations, we use these parameters as these constant parameters to prepare the crystals. This is the batches which we prepared from B1 to B10. This is a drug, is only presented to the EEZ base 4000. It's melted and then converted into the crystals. And then all other formulations were fatty acid base, that is drug in steric acid. And what other variable are? These four are the pore formers presented to the steric acid base. One pore former was of the melt of the batch type, that is a benefit. It's a property is that it's a solid before the body temperature or below the body temperature. And at body temperature, it melts, and that's why there is a formation of the pores into the fatty matrix. And as you know, this P is in the water soluble, so they dissolve into the body fluids and the opposite side, they create a pore. So all four, you can say they are pore formers. Otherwise, it is a fatty matrix having pore formers in different batches. With colloidal silicon dioxide is usually in one of the batches. First one is to increase the viscosity, drug content uniformity. Mostly we are taking the first batch and the 10th batch, B10, where the drug content uniformity was 100%, though others were also tested, and they were having good uniform content uniformity as well within the acceptable limits. So these were the parameters which we measured. Drug content, uniformity, drug radius studies, scanning electron microscopy, we tried to understand the structural characteristics of the crystals, and the stability studies we did by ICH guidelines. Electrical method for in vitro, simple UV spectrophotometer, they use these are the different curves, the standard curves in the different buffers and the different pH media. Mostly we took the SD pH for this because it's oral modified formulation, so it was the stomach pH 1.2 and as well as small intestine pH around 6.8 pH buffer. And these are the in vitro release profiles. You can see the B1, which is a purely you can say immediate release type of formulations, is a PEG based formulation, is a given 100% release almost in 1 hour. Whereas this one is a purely B2, only with lipid carrier as a steric acid having no pore former. So almost very slow release, 20% in almost more than 24 hours, approximately 24 hours. And then when we are using different pore formers, then we are able to, with slight burst effect, we are getting a sustained and controlled release for quite longer duration, that is up to 24 hours. So pastils with IR release, with slow and controlled release having the pore formers in the pastils matrix. This is the drug load. Of course, with increasing the drug load, for example, from 11.4 to, for example, this is the lowest one, having a smaller extent of drug release, followed by the highest load B3, which is giving the highest. So drug load is giving a higher rate and extent of drug release with the increase in the drug load into the formulation. This is the effect of the pore former, especially the benefit, because I said benefit is a specific type of the fatty pore former. And in this case also we have used 3 different concentrations. So you can see with the smaller concentration there is a more linearity and more controlled release. And once you increase the concentration of the pore former, of course the release is more, and also the linearity is a little bit disturbed, especially with the higher concentration of the benefit as a pore former. This is the SEM photograph of B1 batch, which is PEG batch. This is A batch B1, which is a PEG batch, and this B is at higher magnification. And in this case, B10 with pore former benefit. You can see there is the formation of the pores are there once it is kept at 27 degrees centigrade. That is the stability profile, which indicates all the batches are going almost in the rate and extent of drug release profiles at all the temperatures, the storage conditions, and storage times for 3 months. Then, second part was this. We tried to design some more formulations only with PEG base, because PEG is very common and very commonly used, is safe and not very costly as well as it's very compatible to the body. So therefore, only using PEG based, we try to design the 3 formulations, 3 different sustained release, immediate release, and also the pulsatile release. So my focus now will be using PEG bases, having all 3 different types of the formulations. Now this is the batches from P1 to P4, P6. P1 is a PEG, you can say base was 4000 PEG, 4000 milligram, which is a high molecular weight and solid in nature. And there is nothing else. In P2, P3 and P4, just we change the amount of carbon dioxide. And in P5 and P6, So we quoted these particles, especially you focus on the P4, P5 and P6. P4 is entry coat 1, where this is the entry coating material is present with plasticizer. In entry coat 2, we have increased the plasticizer content double from here. And here we have used the floating layer also of the SPMC KF15M over the surface of the entry coat, just to retain it more for the stomach here, so that it can, if you take it into the bedtime, this can deliver to the drug at around 4 a.m. or 3 a.m. early in the morning, as desired by the patient during the asthma attack. So that was the focus. Okay, sorry. So the formulations were all evaluated for these different parameters. So these are the again contact angle measures of these formulations which we have designed for the PEG based pastils. These were just trying to explain that how it's falling on the molten base, is falling on the cooling surface, and then heat dissipation to the cold surface, resulting to the solidification of the lower part and the expansion of the upper part, so ultimately resulting into the shape which we call as pastils. So this is just a mechanism how these pastils are formed. This is the assay and drug content uniformity, which we are getting 100% approximately, and this is the drug release profile. In the 2 hours for the ST media pH 1.2, and for the remaining time it is done for the pH 6.8 pH 6.8 buffer, and as you can say the P1 is releasing within 1 hour in ST media itself. P2 is also releasing, which is not having entry code. P4 is having an entry code 1, which is not very effective. But P5 and P6 is highly effective, as it's not releasing anything, approximately negligible into the stick media, but it's P5 is giving as a immediate release into the alkaline media, whereas P6 having a floating coat is giving a slow and controlled release even in the alkaline media. So depending upon our requirement, we can use this pestle for the use of the bases. And this is the P1. This is P4, P5 and P6. P6, which is having floating coat, is having more uniformity as compared to the other entry coat pastils. This is the floating, which is swelling into the resolution vessel. This is the stability samples. So it is a novel technology. Pastillation was successfully applied for the development of IR as well as control release as well as pulsatile, which I am going to show you next. These are the in vivo studies which we have done, pharmacokinetic studies. Into the VATS, the Ethical Committee of National University has approved the protocols, and we have done it, analyzed it by HPLC, and different pharmacokinetic parameters we measured. This is the calibration curve with the serum, and this is the profile. PEG based is a IR, serum concentration versus time, and this lipid based profile, you can see it's well defined sustained and controlled release serum concentration profile we are getting. These are the pharmacokinetic profile. Parameters Emax is suppressed in the case of liquid based profile, which is desired in the sustained and controlled release. Emax is quite elongated, approximately 7 to 8 times. AUC is high here, and these other parameters are also favoring that this is providing as a sustained and controlled release, especially the liquid based pastilles. And these where this is especially I am interested in this intric with floating coat. This profile you can see almost negligible release are in the blood and serum, and then after that there is an increase, and still it is providing a very good sustained and controlled effect, and concentration is still very high at 24 hours from the P6 batch as compared to P5, and P2 which is uncoated is acting as an IR. So this IR, this sustained and control, this more good sustained and control, and this can also provide the drug desired concentration at the time of early morning asthma attack. That was the basic concept behind this. These are the pharmacokinetic parameters which favors our decision. This is just 1 minute. I'm sorry for this delay. This is the gamma scintigraphic study. We tried to study the location of our pastilles into the GI tract at different time intervals. So we did it by following a specific protocols, and you can see this is the first one where the uncoated PEG pastilles after 0.5 hour and 1 hour it is the stomach. And within 1 hour, you know that you can say the accumulation of radioactivity within 0.5 hours. So that means it is IR formulation. So that's why it is not able to withstand into the acidic medium even into the half an hour. That is the immediate release. And in case of the first coated one, intric-coated PEG pastilles, the P5 batch. This is the location at different time intervals: 0.5 hour, 1 hour, 2 hour. And then this is the last 3 hours. In 3 hours, when it is going into the jejunum, the radioactivity is declining, and then conclusion is the pastilles maintain its matrix integrity till 1.5 hours in the gastric region, and at 2 hours the pastille was located in the jejunum area where it is started to dissociate. No influence of gastric fluid on the intric coating applied on the pastilles. That is what we actually wanted. But when it was a floating coat having on the upper surface, then it was even able to withstand up to 5 hours. So that is what we want it to deliver at the early morning. So it is a pulsatile release type of the formulation. Only in the 6th hour it is radioactivity attenuating. That means the formulation is disintegrating here, or it is losing its property, matrix property. So that particular observation is the pastille was retained in the stomach for 2 hours. In the next hour the exact pastille migrated to the jejunum. Further in the 4th hour the dosage form was found to reach to the ileum region, and then the 6th hour shows complete disintegration of the dosage form. This indicates that protein coat is not only able to retain the dosage form in the stomach for more than 2 hours, but also to protect the injury coat from the alkaline environment. It is also possible. So conclusion is based on all the inferences which I have drawn earlier. That is, the pharmacokinetic and gamma scintigraphic imaging confirms the ability of the formulations to release the drug only after a desired period of time, as is specifically required for the treatment of natural asthma. In agreement, results are in agreement with in vitro drug release and in vivo estimations which are done. And further coating of PG pastille with appropriate polymers can significantly impart the functional properties to modify the release of the drug from these kind of the pastilles. So major findings, we could obtain IR, CR as well as pulsatile release formulations through this new platform technology of pastillations. These are some important references. We have some important publications with good impact factor analysis telling the expert opinion of drug delivery having good impact factors in last 3 years. I acknowledge my student Dwari Shukla because this work is done by her. This is the whole presentation is from her PhD thesis. And this is my group, research group. Some of the people are sitting here who are not included in this because latest photograph I could not take. I welcome you all to a very holy city of the whole world, which we call Varanasi. It is a city of Lord Shiva. These are very famous Varanasi Ghats. So it is this Varanasi University is located in this Varanasi city. This is Lord Mishra. It is said that Varanasi city is standing on the tripods, Trishul of Lord Shiva. So that is a very ancient story. And our educational center is there. This is the BHU, Nashik University, Malviya ji is standing in front of the BHU gate with the founder. There is the Lord Vishnu temple within the campus, which is the biggest of its nature in the world, I should say. Nowhere in the world any educational center has such a big, magnanimous level of the temple. I don't know whether it is required, but it is there. This is IIT campus in BHU. This is my department. This is the entry gate. This is my department where we are working. Thank you so much. I'm sorry for the.
Thank you very much. Unfortunately, we have no time for the questions, so you can ask Mr. Mishra during the lunch break. Another 4 speakers, they did not arrive to the conference, so we will switch to the Young Researchers Forum, and I would like to invite Julia Roe from University.